This is Earth. Currently, about 7.6 billion people live on it. Experts estimate that the carrying capacity of our planet sits at around 9 or 10 billion people, but by 2100, they predict Earth's population will reach 11 billion. However, a recent scientific breakthrough by the University of Illinois and the U.S. Department of Agriculture has shown promise in increasing the Earth's carrying capacity. The researchers have found a way to increase the efficiency of crops by as much as 40%, holding the promise of feeding millions more people. So, how exactly did these scientists manage to increase the efficiency of plants? Through fixing an energy-draining glitch in the process of photosynthesis. Before going any further, let's review how photosynthesis works, and then more specifically, the Calvin cycle. Photosynthesis, which occurs entirely within the chloroplast of plant cells, turns sunlight, water, and carbon dioxide into carbohydrates through two main steps, the first of which is the light reactions. These take place within what's called the thylakoid membranes in the chloroplast. Firstly, water is oxidized, or electrons are taken from it. These electrons are then energized with light energy captured from the sun and used in the photosynthetic electron transport chain to reduce NADP+ and ADP into NADPH and ATP, molecules with high potential energy. These are then sent to the dark reactions, which take place within the stroma of the chloroplast. The dark reactions, also known as the Calvin cycle, take carbon dioxide and in a series of steps, using the NADPH and ATP from the light reactions, create G3P, a 3-carbon molecule which then goes on to form the end product of photosynthesis, glucose. For our purposes, the only step in the Calvin cycle we will focus on is the first one, carboxylation. Normally, the first step in the Calvin cycle involves a reaction catalyzed by the enzyme rubisco. The enzyme adds carbon dioxide to a pre-existing 5-carbon molecule called RUBP, creating a 6-carbon compound which splits into two 3-carbon molecules of 3-PGA. These then move on to the next step of the Calvin cycle, reduction. However, about 20% of the time, instead of picking up CO2 as a substrate, rubisco picks up oxygen. And instead of carboxylating RUBP, it oxygenates it, creating a toxic waste chemical called glycolate. In order to dispose of this chemical, the cell uses a metabolic pathway called photorespiration. Photorespiration, in order to recover some of the carbon lost in the accidental formation of glycolate, puts the waste product through a series of reactions. However, as shown in this picture, these reactions involve an extremely long back-and-forth pathway between multiple organelles inside the plant cell, the chloroplast, peroxisome, and mitochondria. This process wastes energy, decreases sugar synthesis, and some of the carbon isn't even recovered, as it is released again as CO2. Now, back to the scientific breakthrough. What the researchers with the University of Illinois did was test 3 alternate pathways of photorespiration, all which never leave the chloroplast. The 3 pathways were all tested by introducing genes into the tobacco plant, which was chosen due to its fully sequenced genome and short life cycle. In this diagram, the normal pathway of photorespiration is shown in black. The first alternate pathway, in red, relied on introducing 5 genes from E. coli's glycolate pathway. The second, in blue, introduced the genes coding for 2 enzymes from plants and another one from E. coli. The third pathway, in green, introduced 1 gene from algae and another from other plants. All 3 of these pathways were tested in a total of 1,700 tobacco plants, with and without something called RNA interference, which targeted the regulation of the native chloroplast glycolate transporter PLGG1. And what PLGG1 does is it's what transports the glycolate from the chloroplast to the peroxisome, and without the PLGG1, the use of the natural pathways for photorespiration was limited. The results of the experiment found the second alternate pathway to lead to a 10% increase in biomass without the RNA interference. However, with the RNAi, about 24% of the AP2 plants had stunted growth or yellowing leaves. Alternate pathway 1 was slightly more promising, with a 16% increase in biomass without the RNA interference. However, with the PLGG1 regulating RNAi, that improvement disappeared. The researchers hypothesized this was because alternate pathway 1 alone did not have enough capability to handle all the glycolate waste by itself. However, alternate pathway 3 plants exhibited the greatest growth stimulation. Without RNA interference, their biomass increased by 17%. But with the RNA interference blocking the natural glycolate pathway, plant biomass increased between 19 and 37%, with an average of 24%. The tobacco plants using the top-performing artificial pathways had not only more biomass, but developed and grew faster. Currently, the researchers at the University of Illinois are working on translating their findings in tobacco plants to rice, soybeans, cowpeas, tomatoes, and carrots. While it might take over a decade for this technology to be incorporated into food crops and be fully approved, The project successfully realized the potential of photosynthetic efficiency and is working on translating its success into more food for a growing population. However, while this newest breakthrough might again increase the carrying capacity of the Earth, like many innovations before it, it's important to realize that what we're really doing is buying ourselves some more time. So on that note, happy Earth Day. Be grateful for your planet and spend some time outside. But remember, we only have one Earth, and human innovation can only push our planet's physical limits so far.